From a fierce keynote speech, you may get an idea about the landscape of the cancer immunotherapy, the power of it, the promise of it, and the most encouraging results, for instance, the successful utilization of the checkpoint inhibitor, blockade therapy, and the adoptive like CAR-T therapy, and also the in development adoptive T-cell therapy. All those success encourage the development of newer, more potent, and hopefully more affordable immunotherapy for the future. My lab at UCLA, we are particularly focused on the special type of immune cell called invariant natural killer T cells, shortened as iNKT cells. So what are those cells? Why we are interested in them? Consider a drop of human blood. It contains about 10 million blood cells. About 0.1 million of those are white blood cells. Those are the immune cells. And among those, there's about 5,000 of the conventional T cells and about 10 of the iNKT cells. So those are of small number. However, in the war against cancer, if we consider the conventional T cells are foot soldiers, they're mounting the big battles. Those iNKT cells, we would like to consider them as special forces. They are of smaller cell number, however, per cell base, those cells really can respond faster and stronger. So they can make the effect molecules like instantly, like 3 minutes after the interacting of the antigen. And then for per cell base, they can make 100-fold of more of the cytokines compared than a conventional T cell. So all sounds good. So the problem is those cells are of very small number, okay? So in human blood, we have usually the range of 0.01 to 1% of those cells in the peripheral blood. Okay, go back to the nature of those cells. They look like T cells. That's why their name has a T, okay? So they have a T cell receptor, and the receptor has this nature called a semi-invariant TCR, which means the alpha chain of all those TCR are identical, no single amino acid change. And the beta chain is a very limited diversity. So the combination of those make that particular TCR recognize something quite different from the conventional T cell receptor. Conventional CD4 T cell and the CD8 T cell, they recognize protein peptide loaded onto MHC, class 1 or class 2 molecules. Those iNKT cells, they recognize lipid antigen. So they basically cover a whole set of different range of the antigens. And those lipid antigen loaded onto a molecule called CD1D. It is MHC class 1-like molecule, unless it's not polymorphic. So which means every single human being has the same CD1D molecule. Okay, so take that into consideration, and that gives the iNKT cell the capacity to recognize some conserved lipid change. And those lipid change are considered to happen at many disease tissues, for instance, the tumor tissue. You heard from Fier's talk that the lipid metabolism has been greatly changed in the tumor environment, and same, those cells, they can detect those lipid metabolism change. And they can capture those differences, recognize the multiple type of the tumor tissue. And now, because the way it recognizes the antigen, it becomes protein tumor antigen independent and MHC polymorphism independent. So that is one of the attractive features of those cells. And iNKT cells, they are attractive for cancer immunotherapy because there are many clinical indications that those cells are correlated with the cancer, the onset, and the prognosis of multiple type of cancer. In the mouse model, that genetically mutation deplete those cells can cause the animal, in multiple tumor models to develop more severe disease. So we talked about all this. They can target multiple types of cancer, right? And they are independent protein tumor antigen and the MHC restriction. They also are attractive because they can use multiple mechanisms to target cancer. And here shows that for the CD1D positive tumor, the iNKT cells can directly recognize the lipid antigen present onto CD1D and kill those tumor cells. But their most attractive function comes from the adjuvant effect. So the lipid change in a tumor that can be picked up easily by the antigen-presenting cell in that tumor microenvironment. And the APCs, guess what? They always express high level of CD1D that can present the lipid antigen. So by recognizing those signals, iNKT cells can make a lot of pro-inflammatory cytokines that are very helpful for boosting the downstream cascade of the effector cell response, including the NK cell response. In fact, they are the most potent stimulator of the NK cells for in vivo immune response. And also, the iNKT cell can stimulate the dendritic cell, mature them, and by doing that, help the cytotoxic T cell, conventional T cell side of the response. Despite all those attractive features that, as I mentioned, that the limitation is their number. They are of small number in people, and in particular for the cancer patient, that the number is at the really, really low end. So clinical trials has been performed in order to increase the number of those cells to acquire the anti-tumor functionality. So the clinical trial has been performed include stimulate the endogenous iNKT cell through the particular, cognate antigen that has been a lipid has been identified called alpha galactoceramide, alpha galcir. So by injecting that into patient, you can expand the endogenous number of the iNKT cells. However, as mentioned, cancer patients have really no number to start with, so that will not always work for everybody. Another strategy is along a line together with the adoptive T cell therapy. So people have been taking the peripheral blood from the cancer patient, try to massively expand their iNKT cell in vitro, then give them back to the same person. Again, the difficulty is the starting number is really, really low. So we try to tackle the difficulty from a different angle. We use a different strategy through genetically engineer the hematopoietic stem cells. Hematopoietic stem cell is the ultimate progenitor, give rise to all the blood lineage of the cells, including immune cells and including T cells. Previously, we have proved that by delivering a T cell receptor gene into the hematopoietic stem cells, we can program the development of the stem cell, and when those cells go down the path of T cell development, they will go down the path of that particular type of T cells, like we program them to be. So in my Ph.D. study at Caltech, work with Dr. David Baltimore, we proved that in mice, if we give a CD4 T cell receptor to the hematopoietic stem cell, we can successfully generate helper T cells. If we give a CD8 T cell receptor, we will generate cytotoxic T cells. And of course, by choosing the proper T cell receptor, you can give the T cell the antigen specificity you want. And for my interest of cancer immunotherapy, of course, that we have tried to generate cancer-specific helper T cell and cancer-specific cytotoxic T cells. So those are pretty successful in animal model, and our collaborator at UCLA later on proved that it also works for human cells. So you can put a CD8 anti-cancer T cell receptor in human CD34 positive cells and then program those cells to become the human anti-cancer CD8 T cells. So that along that development line, clinical trials testing NY-ESO-1 TCR gene-engineered HSC adoptive therapy, using that to treat multiple cancers, has been ongoing at UCLA Medical Center, led by Dr. Anthony Ribas. So the advantage, a unique advantage of engineering the HSCs, not the peripheral mature T cells, is because HSCs, like the other stem cells, they are long-lived. They can self-renew. So one time of the engineering and adoptive transfer, in theory, can provide to patient a lifelong supply of cancer-fighting T cells. We consider that's an attractive advantage because for a chronic disease like cancer, you need the presence of the cancer-fighting T cells like throughout the life in the person to help check, like make sure the cancer is not coming back. So the question over here is that, yes, we were successful for programming the development of the conventional T cells. Can we do it also for the iNKT cells? In particular, because those cells are small number, they're precious, they're powerful. Can we utilize this strategy to generate enough number of those cells to raise it to the level, the therapeutical level for the cancer patient? So that is my lab at UCLA is focusing on in the past few years. We propose that the HSC-engineered iNKT therapy may provide a new opportunity. Our strategy is clone out of the iNKT T cell receptor gene and deliver to the hematopoietic stem cells. And we hope that will program the cell to become iNKT cells. And not only display the iNKT TCR, we want those cells to really acquire the power of the natural iNKT cell, which means they can respond faster. They have memory cell phenotype, and they can really per cell base, kill cancer and or mount the adjuvant effect at a much larger scale. So in 2015, we published a proof of principle work in PNAS reporting our mouse work. So what we did, we clone out the mouse iNKT TCR. We use a retroviral vector to deliver that to mouse bone marrow stem cells through a very conventional bone marrow transfer approach. We give those engineered cells to a recipient mice. Then we study whether we can detect the generation of the transgenic iNKT cells. So here shows that, yes, we can. And so by engineering step, we can greatly increase the basal level of the iNKT cells that upon tumor challenge mount a much stronger iNKT cell response compared to the control mice. And through the alpha-gal seer immunization, remember I mentioned those cells have the special feature of recognized antigen, and that's a very well-developed agonist lipid already tested clinically to be safe and available to use. So combination with that immunization can really boost the response of those iNKT cells much, much higher above the basal level. So, and in a melanoma lung metastasis model, here shows that untreated mice, you can see all those black nodules. It's a very malignant tumor model. Those are all tumor nodules. So the mice receive the iNKT TCR engineered stem cell transfer. They are much better protected, and in combination of alpha-gal immunization, so the protection is even better. So encouraged by those preclinical study in the mouse model, we assembled a large research team and tried to work on the bench to bedside transfer to move the therapy into clinical trial. So our research team, you can see all those names of those people. It contains 15 UCLA researchers, 3 external board. You may recognize some familiar names over there. In particular, Jim, together with David Baltimore and Steve Foreman, has been sitting on our advisory board. So thank you, Jim, for the support. And this project is supported by a CERN, California Institute for Regenerative Medicine Translational Award. So here is a therapy we're trying to develop. Cancer patients will receive GCSF treatment to mobilize the hematopoietic stem cell from bone marrow to the peripheral blood. Then through local phoresis, those CD34 positive HSCs can be collected. Those cells will be cultured in vitro shortly for 2 days and exposed to a lentiviral vector that encoding a human iNKT TCR gene together with SR39TK suicide gene as a kill switch and as a safety control for the first in-human clinical trial. After that, those cells will be simply collected and given back to the same patient. So it is an autologous transfer approach. And then we'll leave it play inside of the patient. So the engineered hematopoietic stem cells, they will engraft in the patient, go to the bone marrow, live over there, self-renew, stay there, hopefully for the entire lifetime. And their programmed feature will allow them, when the cells go to the T cell development path, which means they become progenitor cell, go to the thymus to make T cells. The transgene will give them instruction to ask the cells go down the path of iNKT cell development, not a random recombinant TCR, not a random Trojan like a T cell path. It's a fixed path now. So we hope, because of the nature of those cells, that they can, as mentioned, target multiple cancer, independent of protein antigen limitation, and independent of MHC restriction. We hope this therapy may have a broader application to treat multiple solid tumor blood cancers, that especially those listed over here have a strong clinical correlation with iNKT cell function. So in order to do that, here is the preclinical animal model that we put together, and we try to make it as close as possible to the human situation, which means everything, all the material has been used are human, okay? So here that we use a lentiviral vector designed for clinical application, and we use a human iNKT TCR gene. We use this suicide control over here. We will use that to transduce GCSF mobilized healthy donor CD34 positive HSCs. And those cells will be given to an immunocompromised animal, many of you are familiar with, an SG mice. And those mice, in addition to receiving the stem cells, will also receive a piece of human thymus to recreate the normal T cell development to allow the generation of an intact T cell compartment in those animals. So those animals will be monitored for the generation of the human iNKT cells. And once those cells are generated, we go ahead to characterize the phenotype, functionality, and most importantly, we study the capacity to suppress the tumor growth. I will quickly run through, some results that we get. It's a pretty large collection of data. Like over here, I want to bring to your attention the upper rows are the non-engineered control BLT animals. And the lower over here is the iNKT T-cell engineered BLT animals. Here shows that the human immune cells generated in those animals at a comparable rate. Human T-cell compartment reconstituted in both animals at a comparable rate. Here is the difference. So endogenously, those mice contain very small number of the iNKT cells, like the human population do. The engineered animal contain much increased population. We can actually manipulate the transduction production rate of the hematopoietic stem cell and control the output of the cells. And for this case, it's probably way too much. This won't be the starting point we run for the phase 1 clinical trial. Here we basically engineered like half of the T cell repertoire to be iNKT cells. It's likely to be an overkill. However, the output can be easily manipulated through the control the gene transduction rate of the input HSCs. So we can, nicely titrate it down to a range, to appropriate range. So here shows the control animal don't have, too many of iNKT cells. The engineered animal, like we detect the, good number of the iNKT cells, and the biodistribution of those cells really resemble what the natural iNKT cell do. They appear in the lymphoid organs, and interestingly, also in liver. And the liver, it is a tissue that in both mouse and human has been indicated to be one of the, tissue like being the homing, a kind of a site for the iNKT cells. Okay, so how those cells look like? Do they really look like the natural iNKT cells? So here is the iNKT cells identified by a monoclonal antibody recognized the invariant T cell receptor. And here is the conventional, T cells. If we compare those both, those two populations, as you can tell, that the CD4, CD8 T cell, those are two distinct populations for the conventional T cells. And the iNKT cells, they show a pretty different pattern. So they show a mixed CD4 single positive, CD8 single positive, and double negative. And that is resembling what happened for the endogenous iNKT cells. And here shows that those cells, the minute they come out from thymus, they already carry this memory T cell phenotype. It's quite interesting. And that is maybe why that they respond really fast. So they respond at a memory T cell, time scale, not really a naive T cell type of time scale. And they also, of course, remember they have an NK name, in their title, right? So they also express the NK marker like CD161 over here. That's also very typical for the endogenous iNKT cell. And let me mention that they do not just express the NK marker. They do have the NK cell function. So they can kill, utilize all the NKG2D, all those, receptor-induced killing the same way as NK cell do. So for killing-wise, they can kill for both, route. They can kill through the TCR, recognize CD1D lipid antigen, and they can also kill through the NK pathway. That's another reason why they can target multiple type of tumor. Okay. Of course, if we want to move this into clinical trial, safety will be the paramount importance, right? So we run those very detailed safety study. We compared the BLT regular animal and the iNKT engineered animal, keep them for really a longer, term, and then check for the body weight, check for the survival, and check for the terminal pathology, the tissue. So the conclusion is that the iNKT TCR engineered stem cell transfer shows no GVHD, no general toxicity, and no tumorigenicity. So it's a pretty good safety profile in this particular animal model. And as you may remember, that I mentioned that we also incorporate a kill switch. So in case something goes wrong, that we will be able to eliminate the gene-engineered cells instantly. So the gene, the kill switch we put is a well-characterized suicide gene called SR39TK. And in addition to be a suicide switch, this gene also can be used as a PET imaging marker. So those cells, you will be able to use PET imaging to track the distribution. So in this case, we use a small animal PET imaging, and it shows that the engineered cells, home to the typical lymphoid organ, bone marrow, spleen. And using a prodrug called ganciclovir, GCV, and the treatment can very quickly eliminate all the engineered cells. Here shows that the imaging, like here, you can see signal, here is gone. And then the results also validated by the flow cytometry, to show that those cells indeed is gone. And importantly, it's a very selective killing, very selective depletion of the engineered cells that we still see that the non-engineered conventional T cells stay intact in those animals. Okay, so I mentioned that the reason that we like iNKT cell because it can employ multiple mechanisms to kill tumor, right? The direct killing mechanism and the adjuvant effect. So we have studied those cells very extensively using various of in vitro culture system, and we showed if we put those iNKT cells together with CD1D positive tumor, they can directly kill those tumor in an antigen and CD1D TCR dependent manner. And when we co-culture the iNKT cell with NK cells, together with a NK target, like K562 tumor cells, that we showed that those iNKT cells upon stimulation can really enhance the response of NK. They like stimulate NK, make them more active and kill their target better. Okay, and when we co-culture iNKT cells together with dendritic cells, we show that they can effectually mature dendritic cell. They upregulate the MHC expression, CD86 expression on dendritic cells, and by doing that, they can help the dendritic cell to stimulate the downstream cytotoxic T cell response. Yet another mechanism quite attractive coming to light in recent years and arise a lot of interest is that those iNKT cells can also target TAM, so tumor-associated macrophages. Tumor-associated macrophages are CD1D positive. So by living in the tumor microenvironment, if they pick up the lipid antigen, can be recognized by iNKT cells, and they will be target of iNKT cells. iNKT cell, by recognizing those TAM, either kill them or suppress their function, that you can suppress the TAM-mediated tumor immune response suppression and help to boost the overall anti-cancer immune reaction. And at last, of course, what it matters most is whether those cells can really kill tumor and control tumor growth. So we utilize a human multiple myeloma xenograft model. So human 1S cells are engineered to overexpress luciferase, as you can see here. So we can track those tumor metastases using bioluminescence live imaging. So here shows that post-injection, those tumor cells really engraft and metastasis, and the distribution is pretty typical, similar to the, disease. So those cell metastases mainly in the bone. You can see then it show up in the spinal of those. So the HSC engineered iNKT cells, by transferring into those mice, can effectively control the tumor growth. Here shows the quantification. And in order to test the robustness of the therapy, that we have tried, the HSC from 3 different healthy donors, and it shows that it worked for all 3. We were able to successfully generate iNKT cell from all 3 donors, and those iNKT cells, are pretty functional and can kill tumor. So up to this point, that I hope that deliver the message that HSC iNKT therapy is a new concept of cancer immunotherapy. We have done proof principle in mouse tumor model, and we are testing this new therapy against various human cancer in humanized mouse models. And we are trying to promote the translation of this new therapy into clinics. And up to this date, I'm happy to report we have arranged a pre-IND meeting with FDA that will take place in early May. So hopefully, with FDA's support, we will be able to move to the clinical stage of development. I'll stop over here and thank my students that who has done those study. And Drake Smith is my graduate student, my first graduate student that published that PNS paper, the proof principle. And our translational team, we call that led by Dr. Yani Zhu, and especially Alice and Victor, to another graduate student. They follow up and really performed the translational study using a humanized animal model. And I would like to thank my many collaborators and my funding support, like to make this work possible. I'll stop here. Thank you for your attention. I'll be happy to answer any questions you may have.
Professor Lili, is a wonderful talk. I have 3 questions. So, first question: do this iNKT cell suffer from on-target of tumor effect, like toxicity to the normal tissue? And second question is: will this tumor escape such mechanism by not presenting such lipid antigen? And my third question is: will an iNKT cell suffer from T cell exhaustion as traditional T cell? Thanks.
Okay, well, those are very good questions. I will try to remember, in order. So you are asking that whether those iNKT cells will cause like bilateral damage to the normal tissue. So the CD1D expression, which is required for the TCR mediated response from iNKT cells, the expression is pretty limited. It's not expressed in normal tissue. So for the healthy organs, they mainly express in APC, antigen-presenting cells, but not the other tissues. Yeah, other healthy tissue express very limited level. And for some cancers, like somehow they upregulate CD1D, probably under the influence of interferon gamma pathway, like all the MHC molecules. So maybe that's why they upregulate CD1D. Okay. So the second question is that the lipid antigen, like whether it can be mutated and then evade the detection of iNKT cells. So it is possible, okay? So our understanding about the lipid metabolism, like lipid presentation and lipid detection by the immune system is much lagging what we know about the protein antigen. I guess that main reason is the technology limitation. Like we're just so good at studying protein. We have genomic tool, we have many other tools. So the lipid antigen is kind of challenging to study. So, but CD1D is a protein that can be tested. And you are right. So like disease like multiple myeloma. So at the early stage, almost like 100% of the multiple myeloma cells express CD1D. But at the later stage, there are reports that at the very late stage, the tumor tend to downregulate CD1D. So that can be a way that tumor get around. So talking about the escape mechanism, I think iNKT cell suffer same way as the conventional cells. However, I did mention that iNKT cell can kill through the T cell pathway, TCR pathway. They can also kill through the NK pathway. So like that's a backup plan, and I guess that's one more layer of control to control the tumor escape. I guess it's been dragging long, and what's the third question? Okay, yeah, the answer is again yes. They are still T cells. They express the exhaustion marker like PD-1, the TIM3. We checked all of those. Actually, they behave similar as the conventional T cell. So that's why we are trying, that's another development path, is we are trying to study the combination therapy. We hope that by combining with the checkpoint inhibitor, and all the other, immunotherapy modalities, like it can really bring up the full power of this therapy. Thank you. Yes, Jim?
Lili, that was a beautiful talk. It's a remarkably ambitious project, and it's wonderful to see how it's come along. I had 2 questions, brief. So the first one is, why do they come out as memory cells? Do they actually see antigen at some point before you're able to detect them?
Yep.
Why would that happen?
So that come back to the development path, and that's something interesting I didn't get a chance to mention. iNKT cell development seems they are the positive and the negative selection stage. It's not carried out by binding to the binding partner on the thymic epithelial cells. They select on themselves. So at the double positive stage, we actually have a figure show that in the manuscript. So the double positive thymocytes upregulate the expression of CD1D. And somehow that provide the selection signal to make those cells mature. And one more information that I can mention is that the affinity of binding. So we all, aware of the conventional TCR when they recognize peptide and MHC. So the affinity is not the same scale as antibody binding, right? That's one reason, like why CAR works that well, because the binding is so strong. The affinity of iNKT TCR binded to a lipid antigen together with CD1D is at the similar scale as antibody binding, not the TCR binding. So maybe, first they have abundant of a signal provided by each other at the double positive stage in thymus, and the second, the affinity of this TCR naturally is much higher than the conventional TCR. Maybe that's why. Makes them a memory phenotype at the beginning.
Okay.
Yeah.
And then the other question I had was, so you, at the very beginning, you talked about the fact that in a drop of blood, there's 10 of these iNKT cells.
Yes.
And so the implication is that there's a very limited amount of space available in the immune system to accommodate these stealthy or super soldiers or whatever. And so when you do this engineering, are you consuming that space? Is there something else that's put at risk, or do you know?
That's a very good point. We think very carefully on that. But let's go step back. If you think about those cells more like a clonal population, they have this invariant, highly conserved TCR. They recognize very conserved, like lipid and a fixed CD1D molecule. If you consider them as a clonal population, they are not a small number at all. When I say that's 10, in your drop of blood, do you have 10 like ESO positive TCR in your drop of blood? And for everybody, right? So that number. And I also mentioned a big range. I mentioned it's 0.01 to 1%. So naturally, for the audience sitting in this room, it can be already 1,000 times difference. And I also mentioned that for cancer patients, their number is at the very low end. Well, that's correlation, causative effect you can imagine of. But how about just simply bring the level to the high end of the healthy population? So there has been report says that that number is around 3 to 5%. So some, I don't know whether lucky or not, but those people just have that high number of the iNKT cells. So it exists in human population. It's safe. So we are trying to design a dose escalation study for the phase 1, and that is our starting point. We're trying to bring it up to matching the higher level in healthy population. And depending on the results, we will decide whether to up or down. Yeah, thank you. Phil?
Yeah. So I also had 2 questions. So
Yeah.
the homotypic interaction, which results in NKT differentiation, does give you NKT cells. But what you didn't talk about is the fact that there are 2 types of NKT cells, one of which is an effector cell, which is great. That's what you'd like. But you're doing nothing to drive it in that direction. There's an NKT regulatory T cell that actually interferes with immune responses, including anti-tumor responses. So what is it that you're doing by putting this in HSC that's going to assure that these cells are actually going to be helpful rather than harmful?
That's a wonderful question. So let me address this question first. Yes, there are 2 types. iNKT1, iNKT2, and the iNKT1 mainly associated with the double negative population and the CD8 single positive population, considered to be pro-inflammatory, and they are like a super killer. They also make the Th1 type of inflammatory cytokine and driving the entire immune response to, a anti-tumor part. And the other side, iNKT2, they express L4 and other molecules, so it's kind of immune suppressive. The simple answer is that we simply put a TCR. We didn't program them to a particular path. But we did observe a very interesting phenomenon. In our animal model, I guess because we are making tons of those cells all at once, and we are able to do time course tracking for those cells, we find that a majority of the cells, iNKT cell, when they first come out from thymus, enter the periphery. If you look at their CD4, CD8 co-receptor expression, which is like correlate but not decisive, point for their functionality, they express this mixed pattern, as I showed you. So like you have cells express, CD4, CD8, or in the middle. However, post-antigen stimulation, especially post a good antigen stimulation. When I see this, take the cells, stimulate with alpha-Gaussian, either in vivo or in vitro. That seems really drive all population, entire population of the cells, down-regulate CD4, up-regulate CD8. So at the end of the alpha-Gaussian stimulation, again, either in vitro culture or in vivo directly inject alpha-Gaussian, those cells, if you catch them early enough, they all are able to go down to the similarly like an iNKT1 subpopulation. So we think that may actually also help to give, in addition to be a translational research, it also helped to give some insight of the iNKT cell biology. So maybe that's what happened in vivo. iNKT cell, when they first come out, they are like conventional CD4 helper cells. They are plastic at that stage. They are full potential. They are not like terminal differentiated yet. So if you generate a good number of those and you can capture that window to give them the proper stimulation, in this case, we are fortunate to have alpha-Gaussian, which is really strong and good stimulator. So if we can combine this with the immunization at appropriate stage, seems that there's chance to really second educate those cells when they come out from the thymus and go down to the iNKT1 path. I hope that may...
I think the risk, of course, is that if you're putting this in an HSC, they're going to keep coming out.
Right.
So you have to either keep giving alpha-Gaussian endlessly, or you're going to run the risk that these cells are going to come out and differentiate along the wrong pathway.
Right. So those are definitely concerns.
And the second question I had has to do with the fact that you presumably are going to have to make space in the bone marrow by depleting, right, in order for these HSCs to graft.
Right.
If you do that, essentially, these cells, when they get to the thymus, because they're going to express this receptor before they do beta chain rearrangement,
yes,
you will essentially be blocking all T-cell differentiation. So will these patients now become T-cell depleted?
Yep, those are definitely good point. And I want to kindly point it out that we can do titration. So which means, when we transfer hematopoietic stem cell, we're not transfer one single cell, right? Or transfer a population of cells. We can easily control the engineering rate by just titrating the amount of virus we use. Like in our experiment, we can titrate, say, we engineer 20% of the HSC. So we program 20% of the outcome T cell repertoire to be iNKT cells. And that, because T cell is about 20% of the total blood, that calculated back to our targeting, initial targeting value, which is about 5% of iNKT cell in the total blood. We can easily bring that down, so we use less virus, so the transduction rate will be 10% 5%, then we will get 2.5% iNKT cell in the blood. If we give more, I hope that's not necessary, then you can have more. Thank you.